Good morning to all. I am Dr. Poonguzhalai, Assistant Professor Physiology Department. And my topic for today's discussion is platelet synthesis and functions. And coming to the synthesis of platelets, and from this picture we can understand the platelets are synthesized from the same progenitor cells which are giving rise to RBCs and WBCs. So the precursor for platelet, it's megakaryocyte, which are coming from the same progenitor cells, that is giving the production of RBC and WBCs. And this is the steps of synthesis of platelet. First, we have pluripotent stem cell giving rise to progenitor cells, and this progenitor cells give rise to promegakaryocyte, and promegakaryocyte to megakaryocyte and the platelet. And the important point from this picture is that the platelet is not the whole cell. It is just the cellular fragments which are getting shedded off from the outer edges of the platelets. So the important thing as a student, what we have to remember is it is nothing but the cytoplasmic fragments which are getting shedded off from the outer edges of the megakaryocyte. And now we'll ask some question and answers. So what is a normal count of platelet? It is 1.5 lakh to 3.5 lakhs per millimeter cube. So this is the total count of platelets. And of these total count, 1/3 of the platelets are sequestered into the spleen. So spleen is having a spleen pool where these platelets are being sequestered. And whenever there is an emergency situation like a heavy bleeding or something, these platelets will be released into the general circulation. So they are readily exchangeable with the plasma. So what is the lifespan of the platelet? The lifespan of the platelet is around 10 days. And what happens to the platelet after this 10 days? The older platelets are killed by the process of phagocytosis by the tissue macrophages which are present in the liver and spleen. So the older platelets are, what is the fate of these older platelets? They are being killed by tissue macrophages. Yes? Okay. Now, this is another diagram again showing the megakaryocyte wherein the platelets are getting shedded off from the outer edge of the megakaryocyte. So these are the clusters of platelets that is getting shedded off. Next, coming to the structure of the platelet. So coming to the structure of the platelet are only cell fragments. They are not the whole cell. So they don't have nuclei and they don't have many of the organelles. They have few organelles like that of the lysosome and all these things, but most of the organelles are missing from the cytoplasm of the platelet. Then what is very importantly present in the platelet? That is our point of discussion. The thing is, cytoplasm of a platelet will have some cytoplasmic enzymes. And those enzymes are very much essential for the production of energy and also for the synthesis of a secretory granules which are essentially present in the platelet. And two types of such secretory granules are present in the platelet. One is the dense granule and the another one alpha granule. And first we'll discuss what is present in the alpha granule. And coming to the alpha granule, they have fibrinogen, von Willebrand factor, platelet factor 4 and platelet derived growth factor. So of these substances, fibrinogen and von Willebrand factor, they are the procoagulant, and these procoagulants are going to help in the coagulation cascade. And what is the role of factor 4? The platelet factor 4, which is present in the alpha granule, will inhibit the endogenously produced heparin that is released into the circulation from the injured endothelial cells. So whenever there is injury, we need the coagulation process to step forward. So all these anticoagulants should be inhibited so that the coagulation process is being pronounced. So the platelet 4 is going to inhibit the heparin that is getting released into the general circulation. And then coming to the platelet derived growth factors. And this platelet derived growth factor, as a name mentioned, it is nothing but the growth factor which is going to help in the aggregation and the proliferation of a myofibroblast at the site of injury, and also help in the repair of the vessel wall. Now the next granule is the dense granule, and this dense granule is having ADP. It's having ADP, calcium and serotonin. And of this ADP is one of the important element which is going to help in the adhesion and aggregation of the platelet. And what is the role of calcium? Calcium, as we all know, it's one of the important cofactor which is going to help in each and every step of the coagulation cascade. And what is the third one? Serotonin. And serotonin is the component which is going to help in intense vasoconstriction, which is essential to stop or minimize the blood loss. And in addition to all these things, within the cytoplasm, we have actin bundle and a myosin bundles. And what is the role of the actin and myosin bundle that is present within the cytoplasm? And these cytoskeletons which are present help in the contraction of the platelet in the process of clot formation. So once it is getting contracted, it becomes strengthened. That's the reason this contraction should take place. And in addition to that, the surface of the platelets will have collagen, the receptors for collagen, receptors for von Willebrand factor, and also the receptors for fibrinogen. And all these receptors which are present on the surface of the platelets have a main important role in the adhesion and the aggregation process, which is essential for the hemostatic mechanism to occur. Okay, and to be very sure, we should understand the granular secretory granules, because these granular substances are only going to help in the whole function of the platelets. The whole hemostatic mechanisms will be regulated by the substances released from these granules. So these two types of granules and the component of the granules which are present are very, very important, and we shouldn't forget that. And the actin myosin filament which is present, it's also going to help in the contractility of the platelet. So with this, I'll go to the next slide. Okay, what is chemostasis? Chemostasis, it's nothing but it is the process of arrest of bleeding, so that the bleeding stops. So before going into the steps of chemostasis, we should understand there is a difference like major bleeding and minor bleeding. Minor bleeding is what is a bleeding which has been coming from the superficial vessels like the capillary venules. And all these minor bleedings are self-limiting. They can be regulated by chemostasis. But the major bleeding, wherein the artery is bleeding, the internal vessels are bleeding, those major bleeding, there is no other option. We have to do some interventions like surgical suturing in order to stop the bleeding. But in case of the minor bleeding, the inner hemostatic mechanism itself is enough to stop the bleeding. So, coming to chemostasis, we have to understand what are the steps of chemostasis. And the first initial step of chemostasis is vasoconstriction. So what is the vasoconstriction? So immediately after the injury, what happens is vessel go for vasoconstriction. And this vasoconstriction is mediated by some chemicals which are released from the endothelial cells. And this vasoconstriction is very much essential to minimize the blood loss. It can only minimize the blood loss, but it cannot prevent the further blood loss. So it is going to minimize the loss until other measures, other hemostatic measures come into action. Yes. So what is the other hemostatic mechanism that is going to help us to prevent the bleeding? That is your temporary plug formation. Yes. So what is this temporary plug formation is our next topic of discussion. So in this temporary plug formation, we have a platelet. And what is underlying your endothelial cells? They are nothing but the collagen tissue which is connected to the connective tissue. So these platelets get adhered to the collagen which is being exposed because of injury. And once this has been adhered to the collagen will activate the platelets. And once this platelet is being activated, it is going to release a substance, ADP especially. We all know that ADP is the where from it has been released. It has been released from the alpha granule. So what it does, alpha granule will promote the adhesion and aggregation of the platelets to stop the bleeding. So, and not only it is getting attached to collagen, this will be attached to fibrinogen, von Willebrand factor, because in this structure we can understand that we have the receptor for collagen, receptor for von Willebrands, and the receptor for fibrinogen. And all these receptors will favor in the adhesion and aggregation in an activated platelet. Okay, and then once it is attached to the collagen, collagen is going to activate the platelet. And this activated platelet, their cytoskeletal elements, what had happened is it gets reorganized to form a spiky process of projection in its outer edge. See, this is the normal platelet as such, and this normal platelet is being converted into a platelet with a spiky projection. And this reorganization will favor the adhesion of the platelet at the injury site. Okay. And this is how adhesion of the platelet is being occurring. And this should be a plug, that is more amount of platelet should be attached to that point of injury. So what happens is an initial plug, what it does is it has four functions. First one is it is going to seal the blood vessel. The first point what I'm going to discuss is this temporary or the loose plug which has been formed will seal the blood vessel. It not only seal the blood vessel, but also it is going to activate the actin myosin contraction process, and this contraction and this contraction process is going to strengthen the platelet plug. And not only that, this activated platelet, what it does is, it is going to release serotonin. And as I've already mentioned, serotonin is mainly released from which granule? Serotonin is mainly released from the dense granule. And this serotonin in turn will favor or reinforce the vasoconstriction which has been created to minimize the blood loss. Okay, so this is how the temporary plaque formation. So what all I have discussed: seal the blood vessel, actin myosin filament get contracted, and it is going to release the serotonin, and that serotonin in turn is going to reinforce the vessel constriction. The last point, what is I want to discuss it is going to activate the platelet, so that is going for the blood coagulation cascade mechanism gets initiated, and that is the process which is going to convert the temporary plaque formation into a definitive plaque formation. Yes, and with whatever points I have told you, so these things, platelets adhere to and they are activated by the collagen at the site of vessel injury. And this activated platelet will release ADP, and ADP activates other platelets, and newly activated platelets aggregate onto growing platelet and release even more platelet attractive chemicals. So this is almost a summary of the whole process of a temporary plaque formation. And now if we appreciate this figure, there is something else is also being explained in this figure. The uninjured endothelium is going to release prostacyclin and nitric oxide. So what is the importance of this prostacyclin and nitric oxide is that the thing is, platelet is getting adhered only to the site of injury. But the normal endothelium, the platelet is not getting adhered to the normal endothelium. Why is that so? The activated platelets, we know what it is going to release. It's going to release ADP. And what it does, this ADP will activate the normal endothelium to produce prostacyclin nitric oxide. And we all know prostacyclin nitric oxide will inhibit platelet aggregation and favor vessel relaxation. Yes or no? Yes. So this is the important point, why the platelet gets adhered to the injured endothelium and it is not getting adhered to the normal endothelium. So the normal endothelium that is being covering the blood vessel or the inner lining of the blood vessel, what it does is it produces prostacyclin and nitric oxide which get released into the circulation and inhibiting the platelet aggregation. That's why the platelet will not adhere to the normal endothelium. This is what is being explained in this fifth point. Normal endothelium or uninjured endothelium releases prostacyclin nitric oxide which inhibit platelet aggregation. So platelet plug is not confined to the site of injury. Yes or no? And now, as I've already mentioned, we have completed vasoconstriction, temporary plaque formation, and this temporary plaque which is being formed will activate the further coagulation cascade to form a definitive plaque, and that definitive plaque formation is the next class which I'm not going to discuss in detail today. So with all these information, what I have enumerated as a story, what are the functions of platelets? It's chemostasis and help in arrest of bleeding. It help in clot formation, help in clot retraction, and it is going to help in the vessel wall repair. In vessel wall repair, which factor is responsible for vessel wall repair? The platelet derived growth factor, which is being secreted from the alpha granule. And told two granules. So from alpha granule, we have platelet derived growth factor, which is being secreted, and that is helping in the vessel wall repair mechanism. And then coming, it have a role in phagocytosis, and it is having a role in the transport and storage. And what it is going to transport and store? It is going to store serotonin and transport it from the organs from where it is synthesized. It just imbibe the serotonin, store the serotonin, and release the serotonin whenever it is needed. So it's helping in the transport and storage of serotonin. Okay, so these are the main functions of the platelets. So for completion's sake, what I have included here is there is injury of a blood vessel wall. So once there is injury, what happens? There is contraction of a vessel wall, and this contraction will promote a plaque formation, temporary plaque formation, and collagen is exposed, platelets is activated, and again that will promote the plaque formation. And the third step, the tissue thromboplastin, which wherein along with collagen, bringing about the activation of coagulation, is the third point, and this will promote a definitive plaque formation. And this part of a flow chart is being dealt as the separate class, and that I will be dealing it separately. And so far, we have completed till temporary plaque formation. Is it okay? And I hope nobody have any doubts. If you have doubts, you can ask me. And that's all for today. Thank you.